wait for the final for the participants that are joining a little bit later, I will go ahead and introduce you. So today we have Dr. Amanda Ziegler. She got her Bachelor of Science in the University of Delaware, and then she decided to move to this side of the United States, and she got her veterinary medicine degree at North Carolina State University in the College of Veterinary Medicine, and her PhD in comparative biomedical sciences in cell biology, also at NC State. And then she moved for her postdoctoral research fellowship, or well, she stayed there, but she got a basic science gastroenterology fellowship from the University of North Carolina School of Medicine. And now she is a faculty of comparative gastroenterology at NC State. And I've seen that you've been doing some work with the microbiome. So
yeah, a little bit. You'll see.
Okay. So welcome. Looking forward to your talk.
Thank you so much. I appreciate that. All right, I'm going to try and share my screen, so please let me know if it goes awry.
Yeah.
Can you see my title slide?
Yes.
All right, perfect. Thank you. Let me arrange my screen before I start. It always blusters me when everyone shoots all over the place. Okay. Thank you so much for the introduction and for the invitation to share how I've progressed with this research since the last time I presented, which was towards the end of my fellowship when I was on the T32 fellowship at UNC. So hopefully I have some more exciting findings to share with you guys. It's building on very much on what I did during my postdoc. So I titled this Age-Dependent Intestinal Barrier Repair in Neonatal Pigs. Because that's the part I'm going to focus on. But in our lab, the comparative GI lab, which I co-lead with Anthony Blickslager, I am a research assistant professor, and we use comparative models, GI models using pigs mainly to study gastrointestinal health across many different barrier-related diseases. But I'll focus on the neonatal repair defect that I've been studying in the past couple of years. All right. So there are many diseases that are associated with mucosal injury across all species in the intestine. So in particular, in swine, which is our model animal of choice in veterinary practice and in mostly in industry production of pork. We have a lot of diseases in swine that are usually diarrheal in nature in younger pigs. And one example of that would be porcine endemic diarrhea virus, which back when I first started my research, killed 8 to 9 million pigs in this state or in this country, and a lot of the piglets in this actual state, because we produce a lot of swine here in this state. In 2013 and 14. It has nearly 100% mortality in neonates, and it's associated with loss of the epithelial barrier and ischemic injury due to the really major inflammation that virus causes. In horses, which is the reason that I do veterinary medicine, I'm a very passionate horsewoman, started out wanting to be an equine vet and then getting hooked on research, so here I am. Strangulating obstruction is it's an ischemic accident in the gut. The intestines of the horse really like to twist, and this causes ischemic injury that causes up to 30% of mortality in adult horses. So it's a major killer of horses, because 1 in 10 horses will colic, and 1 in 10 of those horses will have severe strangulation like this and need surgery. And then 30% of those horses will die despite surgical correction. And then in neonates, it's even worse, reported up to 80% fatality rates. And then necrotizing enterocolitis is an example in humans, which has a high mortality rate and is associated with ischemic injury and epithelial barrier damage as well. Still have reported mortality rates of up to 20 to 30%. And in our lab, we model intestinal ischemia reperfusion injury, or just pure ischemic injury if we want, using surgical modeling in different animal species, primarily pig, The top picture here is actually a horse that we did experimental ischemic injury in. That obviously is extremely labor intensive and expensive to do, but we can do it. This is a pig here, so you can see the scale is a little bit smaller. It's closer to human sized. And then this is a rat. So we routinely see other labs doing ischemic injury in mice and rats. We have done it, but not as frequently. We use these tissues in the lab on Using chambers. Are you guys able to see my pointer? I should have checked. Okay, cool. Thank you, Erin. So this is a Using chamber with mucosa from the small intestine mounted onto it. This allows us to measure, create an ex vivo environment that allows the tissue to repair in real time, and we can measure barrier repair using electrodes and electrophysiology in real time. And it uses this equation here to calculate the barrier resistance, which corresponds to barrier function. And so it's really useful, old school tool that allows us to measure barrier repair in these models in our lab. All right, so we use a pig model, and it's because they have a very similar size, anatomy, physiology, and diet to human. It makes it very fundable. If we find that our studies are very comparable to human, then we're able to leverage the pig model for NIH funding for human health, which is great. But it also is a great model for pretty much all veterinary species. It's an omnivorous animal, and so you can often compare, the gastrointestinal physiology of a pig across many different species. And so as a veterinarian, that's really important to me. In our model, we create surgical ischemia with these ligations most routinely. I like to use the ligations because it really gets pure ischemia, which is a very clean mode of injury, creates a very discrete, repeatable, consistent injury every time. And this is neonatal pig jejunum. So you can see how teeny it is. Those are my fingers holding a length of jejunum from a neonatal pig about 2 weeks old. Then we put it on our Using chamber system for ex vivo recovery, so we can measure barrier function. This is what that looks like when you actually have tissue mounted on it. There's some cell culture media in there from an experiment we did once with cells. So that's why it's pink. It's not normally pink, but that's one of the great aspects of the Using chambers is that you can add things to either side of the tissue so that you can test different interventions in that system to see how it impairs or improves recovery. And so then we can take that tissue after we've measured the barrier function and seen it improve or not, then we can look at the tissue itself using standard histological techniques. And so you can see here, this is tissue from an older pig that has restituted really beautifully. This very thin flattened epithelium here on these shortened villi is indicative of fully restituted epithelium in just a matter of a couple of minutes, maybe a couple of hours. So this is from Dr. Liara Gonzalez, who also works in this area. She made this really great figure in her 2016 paper where she increased the duration of ischemia. And you can see how you can very carefully titrate the amount of epithelial loss. So here's your crypts and your villi of the jejunum, and that epithelial lining there begins to separate, and you get these Grunhagen spaces with just 30 minutes of ischemia. Oops. And if you increase that to 45 minutes, you start to have those burst and rupture. You lose that epithelial coverage on the tips of the villi. And then that damage will, go further and further down into the crypts as you increase up to, I think this is 4 hours of ischemia. So you can really carefully control how much injury you're creating. And the effect of this is because of the way that the blood supply gets to the tip of the villus. So basically the tip of the villus is furthest away from the blood supply. So it's in a relative hypoxic state already, and it's the first to become injured if you were to cut off the blood supply. So that's why it moves in that mucosal to serosal direction. It looks like this when you do scanning EM, and this is a neonatal pig, so the villi are very long because neonates have very long villi at birth. And this was fun because it looks like a turtleneck that's like shrinking down around a shortening villus core. And then this is the epithelial, like Grunhagen's surface epithelium. This space is beneath this, and it's like popping off. It'll probably fall off very shortly. You could kind of see the cells starting to separate from one another. And so I just love this scanning EM image to show that. And when you stain fatty acid binding protein, which is this cytoplasmic marker, actually this might be villain. I better go back and check that. I think this is actually villain because it looks like it's marking the brush border. So I think this is villain, and that's showing you how you really disrupt those that epithelial barrier at the tip here pretty consistently across all these villi. So normally we work with 6 to 8 week old pigs. They have been weaned for about 2 weeks or longer. They're weaned at 3 weeks of age. So these pigs have been on a solid diet for at least 2 weeks. And the reason for that is just because they're a really nice size. It's easier to do the surgeries, and this intestine is pretty close to like a young adult sized intestine. And so that's what we're used to working in over the years to characterize the 3 phases of mucosal repair. So that's that discrete epithelial loss caused by just 30 minutes of intestinal ischemia in this case. And then over the course of repair, what happens are there's 3 phases of what happens here. So first, the villus core contracts down, and that allows the hold on, let me see something real quick. That allows the surface area to decrease. There we go. Which effectively reduces the surface area of the wound so that the epithelial cells that are next to the wound that haven't flown off into the lumen are able to depolarize and crawl across that surface in epithelial wound healing called restitution. And that happens pretty quickly. And then the slowest step is the reformation of the tight junctions, which is how they reseal the paracellular spaces to restore the barrier function. And that is the slowest step. And it is usually complete within a couple of hours in most of our modeling with the older pigs. So, and you can see the new epithelium here. It's not new, so it didn't proliferate. It's just that those epithelial cells right here, over a matter of just minutes, have crawled across the surface here to cover that wound bed. But in previous studies, when I joined the lab, we had noticed that in separate studies in different aged animals that our lab had done, looking at different things, that we saw like really big difference in the recovery of the TER, which is that resistance measurement. Delta or TER is change in TER, but from the beginning to the end of recovery period on the Using chambers. And when we did it with neonatal pigs, we didn't quite get to 100% of recovery. Whereas juvenile has this overshooting, like it doubles the TER well above the control levels in a short period of time. So this was a pretty stark contrast, but we never studied this directly. And it's really important to know if there's any kind of age dependency in repair, because in human infants and also most animal species that we have the literature to review, humans and other babies of all species have worse outcomes if they have gut injury the younger they are. So if our model is showing a defect in repair, then that could be a very important target to study for translational and predictive work in order to improve outcomes in those patients. So this is a human infant tissue from a paper from the Good Lab by Mihi et al. In 2021, showing how much barrier loss there is with human infants that have intestinal injury. This is a neck patient here. So you can see if these epithelial cells can't cover all these wounds, then that's a major problem. So I designed a study when I was in my PhD to compare directly the difference that the impact of age on and basically weaning status on epithelial barrier repair after ischemic injury. And this was the result of that study. So that TER, you can see, starts out lower in both groups than the control, which is normalized to 1 here. And in the juveniles, sorry, I have a magic mouse and it's giving me fits. The juveniles here will increase to the level of control within 75 minutes and then overshoot it, just like we're used to seeing. Whereas the neonates, kill me now. There we go. The neonates have a flat line. They are not repairing their barrier at all during that 120-minute recovery period. And this was due to a failure of restitution. If you look at the juvenile tissues, you see this new epithelium like we're used to seeing, but in the neonates, that defect after recovery is persistent. So there's still many villi that don't have, you can see maybe it's attempting to here, but what is happening? Hold on. Is there like a auto change function that I've turned on? Does anyone know anything about something like that? Because that's not me.
Sometimes you can set a timed presentation so that it just continues to advance.
I hope it'll stop. I don't know how to stop it. I don't normally have snafus. I'm too. Oh man, this is embarrassing.
Is it self-advancing the slides?
I think so. Do you know how to turn it off?
Yeah, you have to go and turn on. You have to exit out of the presentation, and then go in and turn off the timed version.
It's on the transitions.
Okay.
Part.
All right. Oh man. On mouse click. Oh, I found it. Oh my God, thank you guys. That was awful. Oops. I think I fixed it. Here's hoping. Does it apply to all slides? Oh wait, here we go. I found a button that says that, so I'll click that. Okay, here we go. Here's hoping. Okay, so here we are. So we have this persistent wound that persists in the neonates. It's not recovering, and this was quantified easily, showing almost 100% recovery in juveniles and actually worsening of that wound size in neonates. So this was due to a complete lack of this epithelial restitution phenotype in these pigs. So they have these epithelial cells here. This is the wound bed here marked with the asterisk, and this is the subepithelial tissue here. These are epithelial cells that are depolarizing and flattening to crawl across the surface on SEM. So you can see those little microvilli are like being recruited, that redundant membrane is being recruited into this advancing lamellipodia. And it looks like pancake batter oozing across the surface, and it's very cool. But in neonates, it looks strikingly different. So there's no recovery at all. Those little tennis balls are just sitting there furry, not doing anything to cover that wound bed that they're sitting next to. So that's a major problem. So when we did RNA sequencing on this, on these tissues, it's just homogenized whole mucosa, in this case, bulk sequencing. It is showing that there is a stark difference between this transcriptomic profiles of the juvenile pigs here versus the neonatal pigs here. And then when we break this out with ischemic injury, let's go over here. So this is ischemic injury comparing neonatal versus juvenile whole mucosa transcriptomic profiles. And you can see here, this is the juvenile, and they have a really tightly organized, precise response that they're all doing transcriptomically. That's very different from these neonatal tissues, which have a more spread out transcriptomic profile from one another. And it's different, very much different from the juvenile. So that would suggest there's definitely some sort of signaling differences among these different age groups. It's very weird. So the cell crawling pathways, this is ingenuity pathways analysis on that RNA sequencing data for diseases and functions related to anything I saw in this that was related to cell crawling. And if I looked at any of those that are, cell movement, migration of cells, organization of cytoskeleton, cell protrusion, these kinds of things are all downregulated in the neonates. So it confirms our phenotype for sure. All right, and then in the case of GSEA analysis, we saw similar things with the, pathways that are gene sets that are relevant to epithelial barrier function, or restitution. So apical junctions would be important for forming that those tight junctions that maintain the barrier, and that's downregulated in neonates. This inflection of the GSEA plot would indicate that this is enriched in juveniles and as opposed to in neonates. And then the integrin cell surface interactions, which are like the adhesions that would allow them to crawl across the surface, are also enriched in juveniles and absent in neonates. And then interestingly, this EMT pathway was downregulated in neonates as well. And so that would be similar to, I'm very interested in this EMT pathway, and I'll get into it a little bit later towards the end. about what I want to do about this, because it's very interesting to consider that there might be EMT-like signaling going on that's driving that depolarization and crawling behavior that's very similar to what you'd see in mesenchymal transition in cancer and development and stuff like that. So corroborates what we're seeing visually. So we wondered early on, with the help from my mentors in my T32, this is actually part of my T32, whether these cells could actually crawl. And we thought, well, if we were to take all of the signaling that's happening in the juvenile animals and just place it on the neonatal tissues, would it force those epithelial cells in the neonate to repair? Can those epithelial cells respond to that milieu from the juvenile? And so we did it. It was goopy and it worked beautifully. So within a few minutes, we had a really big increase in the tight junction structures and the barrier function with that juvenile homogenate, but not a controlled neonatal homogenate. And in those tissues, when you look at the juvenile homogenate treated neonatal tissue, you can see more of these epithelial cells that have restituted the villi. Very cool. So, but we don't know what's doing it. It could be anything in that homogenate, because we just took the entire surface of the mucosa and just homogenized it. So it could be something related to like mucins or microbiome or nutritional inputs, and it could be anything from underneath as well in the set, the subepithelial tissue. So that's where we've been going in recent years, trying to understand where that's coming, what is driving that improvement. So we work a lot with Dr. Van Lendegem, who I think most of you know very well. Her lab is adjacent to ours at the vet school, and she pointed out to me when I presented this early on that the subepithelial network of glial cells, part of the enteric nervous system that she's an expert in, develop postnatally, and they promote barrier function by paracrine signaling. So those epithelial cells that are close enough to the epithelial cells could actually improve restitution. Here's a paper that she published back in 2011 showing that in vitro. So if you co-culture cells with glia, they restitute faster in wound healing trials in vitro. And so we looked in our tissues and found that when we did GSCA analysis for glial development relevant pathways, it's all enriched in juvenile tissues versus control or neonatal tissues. So we have cell migration, proliferation, development, and differentiation all enriched in the juvenile tissues. We looked at this visually using iDisco. I've shown this before to you guys, but I'm showing it again because it's fun. But this is just 3D imaging of GFAP signal, which is the primary marker of very active enteric glial cell subpopulation in paracrine signaling. That's the neonate, and you can see the subepithelial, the epithelium is all the way up here. So the subepithelial glia are going to be right here in that mucosal tissue. But then in the juveniles, you can see there's a lot more staining in the subepithelial tissue. You'll see it better once I isolate one villus and show you just one villus core filled with glia versus this one. So we quantified this and found that there's enriched density of those important GFAP positive glia in the juvenile tissues versus neonates. Which would support that they might be important for driving repair potentially. So when we looked at, recently I've looked at a paper came out in 2021 from Schneider et al, where they defined enteric gliosis, which is inflammatory signaling in glial cells. That's just the term that it's been given. And this is a gene set associated with gliosis. And it's just an activated state, usually associated with inflammation, but I would imagine it could be activated by ischemia, any kind of injury. So I was curious about whether that would be enriched, and it is in the juvenile tissues. And my student, Maddie, helped me with this study where we took fluoroacetate, which has been shown to inhibit glial activation, glial activity, by blocking, they're more sensitive to it. It's a TCA cycle inhibitor, but they're supposedly more exquisitely sensitive to it than other cell types. So you can titrate it to get inhibition of glial activity only. And she found that she could treat this with 500 micromolar fluoroacetate and get reduction of that induced GFAP, which is a part of the gliosis response. GFAP is one molecule one protein which could be upregulated during gliosis. So at least in the case of GFAP, she seems to have been able to block that increase in response to ischemia in our pig tissues. And these are subepithelial or submucosal plexi here that we've used as markers. And so when we took this inhibitor and we used it in vitro or in vivo and ex vivo, actually, we put it in the pig loops as well as in the Usain chambers themselves. Any dose of fluoroacetate will block recovery of TER versus control. And then when we look at the epithelial injury, the only dose that doesn't cause actual epithelial injury by itself without ischemia, but is effective enough to block repair of the epithelium is the 500 micromolar fluoroacetate dose. But we were able to effectively inhibit repair, and it looks like this. So these epithelial cells here have failed to restitute, and this is due to being treated with 500 micromolar of fluoroacetate. So that would suggest that inhibiting glia would inhibit repair in our juvenile pigs. These are juvenile pigs, which normally can repair. And we have a lot more work that we're doing currently because this is in preprint with AJPGI. And we have some work we're doing to try to show better, more thoroughly that this is inhibiting only glia. So that's in progress. And then when we take glial cells out of the tissue, these pig glial cells, we can culture them. Lorianne has helped us to adapt the protocol that she uses to our pigs, and we can routinely culture glial cells to study them in vitro. And so this is just a representative image of some GFAP and S100 beta positive glia, which is a very common subtype known to be involved in paracrine signaling with the epithelium. And we put those cells in a transwell system with IPEC-J2 cells, which are a intestinal pig epithelial cell. It's a jejunal pig neonatal epithelial cell line that we then scratch wound and can see that the submucosal plexus glia and the longitudinal myenteric plexus glia both enhance restitution rates in those cells in vitro. So that further supports that this might be important for our model, this glial signaling. And then I just wanted to take a little sidestep into and just kind of scratch the surface of this project. This is a very complex team science project that we've got going on in the lab, which is relevant to this whole body of work, but is funded by the USDA to study how the glial network is involved and how nutrition and microbiota might be driving maturation of the gut and barrier function repair. So we know that the glial network completes development in response to postnatal microbial colonization of the gut, at least in some animal models. It's been studied mostly only in rodents, and people debate all the time about whether this is true in humans. It's a very new field. But in this study in rats, you can see that germ-free mice don't have those subepithelial glia in the mucosa. But if you inoculate them with the conventional microbiota, you can rescue that and actually produce it. So it would seem that it's responding to the microbiota in order to go into those subepithelial spaces where they would then be close enough to interact with the epithelium. Our collaborator, Jack Odell, is in the animal science department at NC State, and he is a neonatal nutritional biologist. And he has previously shown that microbiota can be modulated in pigs perinatal pigs by dietary prebiotic interventions. And so you can see these microbiota, I forget what this analysis is, but it's showing the diversity of the microbiota in the pigs is clustering separately if you've had prebiotics added. This green and this purple group both had prebiotics added, and they're clustering distinctly by day 21 of age. And so together with Dr. Lorianne, Dr. Blixlager, and Dr. Van Landingham, we came up with this idea that we put into the USDA that dietary oligosaccharides, prebiotic fiber, which feeds the microbiota, would accelerate neonatal microbial colonization and thus glial network maturation, and thus enhance restitution responses in neonates after ischemic injury. And so in this study, we've been working on this for 4 years. It started just before COVID, so it's been a little it's been a little delayed, but we've been working through it, and we have a lot of data to work through. But I'm just going to show you a sampling. We took piglets that were fed just 24 hours so they could get their colostrum from their sow, the first milk. And then we put them in these animal health building auto sow facility that has individual cages with little nipples going into the cage so that you can carefully titrate exactly how much of what kind of formula each piglet gets. And it's really useful for testing different nutritional interventions. So they either got prebiotic supplemented replacer or just replacer. And replacer is a animal production word for formula. So it's the same thing. And then we, at 1 day, 7 days, 14 and 21 days, we took those pigs for microbiome analysis, glial network development assessment, barrier function assessment. And then at 14 days of age, which is that age where we previously characterized this major defect in repair, we tested glial function and then also epithelial barrier injury and repair to see if we've impacted that with this fiber. And so these are just some of the results. We have to sort all this out, but it's very interesting. And the short version is that the prebiotics do affect everything, but trying to like work out how has been a long road. But this is basically, these are showing the diversity of the microbiota of these pigs at 7, 14, and 21 days of age in the colon. And we've switched to colon here. So just to be clear, the low and high prebiotic diets are here versus control. So at 14 days of age, we've shifted the diversity, but then it kind of, there's still some separation, but it's mostly resolved by 21 days of age. So it seems like there's a shift in the early microbial taxa at that important time point that we're studying. And then interestingly, we got the opposite result of what we thought. And there's actually reduced GFAP with the treatment groups in the mucosa. This is whole homogenized mucosa from these pigs, and it's reduced in pigs that were treated with prebiotics at 21 days of age. And it was only significant, there was only a change at 21 days of age. And then same is true for S100 beta, which is another marker of glia. And so this is strange, but interesting and very significant. This is some preliminary images of the iDisco volume imaging that we're doing to try to characterize the changes in the glial network development in these pigs, and it's still early. We have a student working through those now. It's a lot of work to work through all these analyses with the three-dimensional imaging technology. But this hopefully shows you a little bit that there's some difference in the distribution of these markers in that subepithelial space here. I won't spend too much time on that. She hopefully will have that finalized soon. And then Lorianne took these, it was a flurry of culturing activities. Every time we did these trials, every summer she would be working, culturing many glial cells from all these pigs all at once in her lab during the summer with help from her whole team and some of ours too, where she found in vitro that the cells cultured from these pigs, from the colons of these pigs that were fed prebiotics at 14 days of age, had differences in their proliferation rates if they were from the mucosal plexus, which is the outer plexus. There was no difference in the submucosal plexus. And in the submucosal plexus, there was a decrease in response to ATP, like calcium signaling responses to ATP in the submucosal plexus cells, but not the lamina-propria or LMMP cells. And then they did a really cool migration assay where they tested the chemotactic ability of these cells in Boyden chambers and found that in the SMP cells, the submucosal plexus cells, there's reduced chemotaxis in the treated, in the glia from treated pigs. And we did save the supernatants from these glial cells in culture, and she has submitted them for mass spectrometry and to look for what the secretome profiles are how they differ and might be driving any changes in paracrine signaling going on. And there were 13 proteins of interest that she had told us about, and one of them was thymosin beta 4, I believe, and that's been implicated in epithelial wound healing in other systems like the skin. So I'm interested to hear what becomes of that, and that's still in progress as well. And then when we culture these glia from the pigs that were fed prebiotics from the colon. The prebiotic fed colonic, so basically the control does a pretty good job already of enhancing repair, but it's a little bit better with the prebiotic fed in the submucosal plexus cells. So it's very interesting. It's a subtle change, but I think the main takeaway from this that excites me a lot is the fact that this is all being retained in vitro after a couple passages. So like this is just feeding pigs prebiotic fiber, which is a very simple intervention. And it's drastically changing the physiology of cells in culture several passages down the line. So I think that's a really strong phenotype. And we're really excited to keep studying this. So for this study, our conclusions and ongoing work are that we know that prebiotics will induce changes in the microbiota and glial cell activity in neonatal pigs. It increases epithelial restitution in the small intestine. I didn't show you that, but it's a very subtle change. And I'll just leave it at that. Increases the, this is colon. I meant to say colon, so I didn't fix this slide, but it increases glial-driven restitution in vitro from colon cells. So, and then ongoing work with the iDisco will fully characterize that glial change network in 3 dimensions, which will be really cool. And then we have lots of ongoing co-culture studies that will specify specific signaling mechanisms. And I'll give you just a little teaser because I don't want to steal Maddie's thunder, but this is Maddie Caldwell. She's working on this project for her PhD where she's studying how those glia are prompted to actually induce repair. So what is the signal that the glia are getting in order to tell the epithelium to repair? And that's her area of interest. So she created this really creative experiment with Lorianne Van Landigen's help, where they are taking cultured glia from pigs. And in this case, she took them from neonate and adult pigs and cultured them with IPEC-J2 cells and then scratch wounded them and looked at the percent closure at 8 hours after wounding. And this is a bit of a complex graph, but what I want you to take away from this graph is that when you add the luminal contents from a pig that had been sterile filtered to the culture medium on the apical, in the apical chamber above the IPEC-J2 cells, just before you scratch it, the effect of those luminal contents on restitution of those epithelial cells depends on the presence of glia. So in the absence of glia in monoculture, it actually decreases epithelial wound healing, but in the presence of glia, mainly neonatal glia, it will increase wound healing. And so that's detected by this interaction on the ANOVA between luminal content treatment and the co-culture. So I thought that was really exciting because it shows that those glia are having a really important regulatory role in how the luminal environment impacts restitution of the barrier. So I'll leave it at that. She wants to present this, I think, this summer. She's going to submit this for a poster soon. So. And then I just wanted to circle back to the EMT. I told you I wanted to get into what this EMT stuff is all about. And so I've recently submitted a couple of R grants. One of them is an R01, and I'm hoping that everyone will share my enthusiasm that this EMT thing is really cool. And I want to define the restitution reprogramming that's happening in these cells. So that's the target of my upcoming pending proposal. So in these epithelial cells that are restituting in the juvenile pigs, what is actually happening? It's not all that well defined. It's been defined a little bit here or there, but no one's really looked to see exactly what these cells are doing. We know that it involves actin rearrangement, depolarization, lamellipodial extension, integrin adhesions, things like that. But as a whole, I want to know what those epithelial cells are doing. And so in the recent history, we've sequenced those cells. We took those cells and sloughed them and suspended them into single cell suspension and generated libraries for single cell transcriptomic profiles. So these cells that you're looking at here is a very preliminary UMAP of just 1,359 cells that managed to survive this whole ordeal. They were injured. This is pig mucosa that had been injured by 30 minutes of ischemia and then recovered ex vivo for 30 minutes. And then we took those little teensy weensy tissues off the chambers. And Elizabeth actually is doing this. Elizabeth, I forgot to put her picture on the slide. I'm so sorry. If you're here, Elizabeth, I'm so sorry. But Elizabeth is doing this work, and she sloughed all these epithelial cells off and then submitted 8,620 cells for sequencing. We had 1,359 cells of adequate quality to analyze. And so that's what you're seeing here on this. There's not very many cells, but they're very injured and sad. But we're hoping that within these cell clusters that we can figure out which of these is a restituting subpopulation, maybe a subcluster. And so I've been looking at this just this week. So this is hot off the presses. I'm trying to figure out how to identify these cells. And I've been looking at the paper from Joseph and Scott and his whole lab that characterized the individual transcriptomic profiles of all the epithelial cells in the human. And I'm trying to figure out which is which. And based on what I've seen from their publication, I think this is an absorptive enterocyte population, and this is most certainly a tuft cell population, and this is most certainly an enteroendocrine cell population. This is, I think, best 4 cells, which pigs have and mice do not, and humans do. And then I think this might be transit amplifying cells, but these are all kind of a mystery. This follicle associated epithelium, maybe, but these 4 here are kind of a mystery to me. So I need some help I'm going to work with Bark to try to sort out what these are. But when I'm looking at the clusters, and they did a gene ontologies analysis of these clusters, and these are selected gene ontologies that are enriched in cluster 0 that I think is absorptive enterocytes. And there's a lot of stuff relevant to plasma membrane, brush border, apical response to wounding came up as ranked 40, which caught my attention. But cell projections, things like this. So maybe that's an interesting cluster to look at. And then weirdly, this is the one that I was most excited about, because ranked high up here, 2, 3, 6, 8, are things related to cell projections, cell motility, lamellipodium assembly, that's really relevant, locomotion, cell migration. So this profile here is getting me excited, but they're tuft cells. So that's really weird. I mean, I'm pretty confident these are tuft cells based on their profiles of enriched genes. So I don't know, I need to work on this. So I'm just going to leave it at that. I just wanted to show you where I was with that bit of the project, because it's the most exciting part of what's going on right now. And then as a sidebar, we also have a project where hopefully it'll help me work out what these clusters are, is to do a full survey of the pig GI tract using single cell sequencing, very similar to what the Magnus lab just published with Joseph's immense help. Hopefully we can sequence the pig as well, and then that'll help us work through these questions. And with that, I will stop. I think that is a good amount of time and enough data to share with you guys. So I would love to take any of your questions that you might have, or suggestions. Love suggestions.
Thank you, Amanda. That was very, very exciting and very interesting. Do we have questions?
I have a question.
Yeah.
Oh, hey, Amanda, that was fantastic. About with your single cell RNA-seq work, I'm curious, you did it off the leucine chamber. I guess you damaged it. Did you do ones that were not injured, just to sort of get like a more of a healthy profile before you dived into the more diseased aspect of it?
We haven't, but we are right now. So there is a project about to start at any moment where we will slough healthy epithelium from distinct regions of the GI tract from the pig, and that'll give us a lot of data about the jejunum in a normal condition. And I think it'll really help us work this out.
And just one more question. So the glial cells, you're setting up co-cultures, but the cells, the glial cells are primary, but the co-cultures are being set up with a cell line, not with another primary cell, right? From the same pig. Okay, correct. Are you planning to maybe try to venture into the? Yeah,
we have done monoculture of primary, basically 2D aneroid culture. Right. It's a little harder to get a consistent monolayer that scratch wounds and restitutes quickly. I know Erin's done that quite a lot recently. We want to do more of that. And so that's kind of where we're going now. Most of our historical data was in IPEC J2 cells, but I'm pushing for more and more primary because that hopefully is going to be more representative of in vivo.
Excellent. Thank you.
Yeah, Amanda, that was an amazing talk.
Oh, thanks.
I have a very basic question from like the first slides of the background. I was
Okay.
struck by the sheets of cells coming off during the early hours of the injury. I forget the name of like the bubble that forms below them. Do you know, it looks like there's just too much epithelium to cover the stroma at that point. Is the stroma in the middle of the villus actually shrinking? Or are the
Yeah.
cells kind of artificially pushing up and getting pushed off? Do you know what's going on there?
It is, yeah. So the core of the villus, under mostly action by the enteric neurons and prostaglandins, is what we understand it to be, are acting on the smooth muscle fibroblasts in the muscular core of the villus to actually contract it down. So you're right. And that's that SEM image I showed you where it looked like a turtleneck, like shrinking up like this, is a great example of that. So it shrinks down and it kind of like gets much shorter and it pops off that epithelium, I think, more aggressively than if it was not doing that.
And then a follow-up question with that is, do you know if there's changes to the stromal cells itself? Are there like adhesion proteins that could be lost and could be different between neonate and juvenile?
It's such a good question. The only one I've looked at is the basement membrane. I stained it with collagen type 4, I think it is, and it looked similar across groups, but I haven't explored that further. It's a really good idea though, and could potentially be really relevant because that subepithelial matrix that they're adhering to could be vastly different between the different age groups. Yeah.
Cool. Thanks for the great talk.
Thank you.
I have to ask you about the prebiotics experiment. So we work with prebiotics. We work specifically with galactoligosaccharides. So can you tell me about the origin, the purity of the prebiotics? And then the pig that received the prebiotics, did you look at the alpha diversity? Like the diversity in the samples, or like how many species were in the ones that received the prebiotic versus the one that didn't?
So for your first question, the makeup of the prebiotic concoction that we used for our experiments was 8 grams per deciliter of a 50-50 mixture of galacto-oligosaccharide and polydextrose. And it was created for Dr. Odell by the nutrition company that he works with extensively. And so it's a swine nutrition company. And so the purity, I would have to ask him for details, but he routinely uses their formulations for his research. And so I believe there would be stringent quality controls for the purity. And then your second, yeah.
No, I don't doubt that. It's just that normally, you know, commercial gauze has about 60% gauze, the rest is lactose, glucose, other sugars mixed up. That's what I meant. Not about the quality.
I just don't know. I just don't know the answer, so I just wanted to say I'm pretty sure it's good.
No. Yes, of course. I have no doubt. Yes.
And then your second question was, if we looked at alpha diversity, and yes, we have a lot of data that I did not show, pairing the different groups. I just showed like one that was easy to explain because I am not a microbiologist. And it's all there. And I actually would love help looking at it when we go to write this up. So if you don't mind, I might even reach out to you to help me.
Absolutely. Happy to. That's what we do. That's what
Perfect.
That's what my question. Yes.
Thank you.
Sure. I think Ian.
Yeah, yes. Hi, Amanda.
Hi.
Very good talk. It's a lot of very interesting stuff. And I guess I have probably a pretty basic question for you in terms of how localized ischemic injury is. Do you only see injury in this segment of gut that is actually blood flow is restricted to, or do you see changes? I'm saying like gross physiological changes in villi size upstream or downstream of that segment?
That's such a good question. So we actually have Emily Hellstrom in the chat here, who is studying the impacts of just intestinal manipulation on barrier function in these pigs. And her interest is in postoperative ileus because she believes there's a link between early decreases in barrier function and the inflammatory phase of postoperative ileus. And so that's what she's studying. And in her models, she's actually ramped up the injury in non-ischemic adjacent tissues by creating ischemia, because she found just manipulation alone was a very subtle change. But if she created a more clinically relevant injury like ischemia, then the adjacent tissues got a lot more permeable. And so as far as the more like grossly more like histologic changes like villus length or anything like that, we haven't looked at that specifically. I don't think she's measured that specifically, but the barrier function is definitely decreased in the adjacent tissues. It's just not to the same extent, and it recovers a lot quicker. And so that's probably just from tissue handling inflammation, just signaling related to trauma to the tissue during surgery.
Yeah, very interesting. Thank you.
Mm-hmm. Oh, and I should also add, she doesn't see any epithelial loss. So without the ischemia, there doesn't seem to be sloughing of epithelium, just leakiness.
Okay, great.
And this is small intestine, right?
That's right.
Interesting. Any additional questions? If not, thank you so much again for a very exciting talk and for answering all of our questions.
Yeah, no problem. I love the questions and the suggestions. I really appreciate your input. Thank you so much for listening.
Okay, thank you.